Hello everyone! Thank you so much for joining me for today's Genes in Space live webinar. I am Dr. Alex Dainis and I am so excited that I get the chance to tell you about this amazing competition for students in grades 7 through 12. So if you haven't heard of the Genes in Space program before, Genes in Space is an amazing contest open to students in grades 7 through 12 in the United States. So students can work individually or in teams of up to 4 students, and you can think of it a little bit like a reverse science fair. So in a normal science fair, you would do a bunch of experiments and then write them up and then compete. But instead, for Genes in Space, all you have to do is have a great idea. As long as you have a great idea, you don't need to do any experiments. You just get to propose and design an experiment that might be done by astronauts up on the International Space Station. So we have been running the Genes in Space contest for about 5 years now, and we've sent 6 experiments designed by middle and high school students up to the International Space Station that have been performed by astronauts and that are really pushing molecular biology in space forward. Now, each competition application has to be sponsored by an adult educator. And one of the fun things is that if you win, if your experiment is chosen as one of the winners, you actually get to go to Cape Canaveral with your teacher to watch it launch to the International Space Station, where it will be performed by astronauts. So this is a really, really cool and very unique contest for students, and it is completely free to enter. And this year's applications went live in January. So you can write up your proposal and enter right now. So why are we asking students to write in proposals and experiments about space exploration and space biology? And one of the reasons for this is because we are entering a new era of space exploration. So you may have heard about missions that are going to try and go to places like Mars and beyond, and we want to know what's going to happen to those astronauts when they go to space so that we can be prepared for it. So we already know that when astronauts go to space, their bones start to weaken and their muscles start to atrophy. And we want to make sure that if somebody's going to Mars, they're not losing all of their bone and muscle mass. Now, because we've learned these things from sending astronauts to the International Space Station, we were able to put countermeasures in place. So astronauts who are up there right now take additional dietary supplements, and they do up to 2 hours of exercise a day to keep their bones and muscles strong. So the whole idea is that the more we learn now, the better equipped we can be to go out into space and to tackle these problems before they become big issues. Now, one of the great things is that we have the International Space Station as a testing bed for all of these questions. So if you are like me, before I started working with Genes in Space, I didn't really know what happened up on the International Space Station, but it turns out that it is basically a giant orbiting lab, and the astronauts do experiments for people all around the world. So they're running experiments from places like NASA and the European Space Agency. They're also running experiments from places like universities, but additionally, they're running experiments designed by students just like you. So you can design an experiment and have an astronaut perform it up on the International Space Station. And this is really cool. I mean, the ISS is one of our only microgravity facilities on Earth. It's not actually on Earth, it's up in space, but it's one of our few testing beds where we can actually look in low Earth orbit at what might happen to astronauts and other organisms when they go on these much longer journeys. So one of the really cool things about the Genes in Space program is that you might notice that there's a little machine in one of these pictures called a mini PCR, and there's one right on my bookshelf behind me. So this is a miniature PCR machine, and Mini PCR Bio is a sponsor of the Genes in Space program. And it was in fact the first PCR machine to go to space, and the first PCR, or polymerase chain reaction, which is a really critical molecular biology technique that was performed up in space, was designed by one of our winners, an amazing high school student named Sophia. So Sophia designed an experiment that we'll get into a little bit more in a minute, but she really pushed forward the ability to do molecular biology up in space. Without Sophia's project, none of the other experiments that have come after it looking at molecular biology and doing things like DNA studies would have been possible. So it's really cool that all of that came from a high school student's idea and proposal and experiment through this contest. So there are a lot of challenges that we know about when astronauts go to space. So for example, we know that when they reach these microgravity conditions, we'll see eye and optic nerve compression in these astronauts. So this can lead to vision problems both on space or while they're in space and afterwards. We also know that they sometimes experience bone density loss, as I talked about before, and because we know this by watching these astronauts go to the International Space Station, we can come up with better ways to combat it for astronauts who are going to go out into deeper space. And we also know that our atmosphere down here on Earth protects us from UV radiation from the sun that can actually cause permanent DNA damage and other types of radiation. But we know that when we go up in space, we lose that protective shield of the atmosphere and we're exposed to more of these damaging rays. So one of the other things that we want to be able to study is how our DNA is affected by being up in space. And you may have heard of the twin study with Scott and Mark Kelly that looked at how spending a year in space affected all kinds of human DNA. And there are lots of human-focused questions that we can try and tackle with genetics. But humans aren't the only living things on the space station. So there are also lots of microbes. So you and I are completely covered in bacteria. And when we go to space, we bring all of that bacteria with us. So we know that there are bacteria that live on the International Space Station, and we want to see how they grow and change in microgravity. We also might want to be able to use genetics to study if an astronaut gets sick in space, to have a much faster way of diagnosing what they're actually sick with. Because if you're on a 6-month-long mission to Mars, it's going to be pretty hard to try and take a swab and send it back to Earth to get an idea of what you might be sick with. You might want to do some DNA sequencing to find out a little bit faster. But you'll notice there's also a plant in this picture, and this is one of my sort of favorite topics to tackle, thinking about living organisms in space, is plants. So if you are on a 6-month mission to Mars, you're probably going to want a fresh vegetable at some point in time. So there's a lot of work going into figuring out how we can grow better and tastier and more nutritious food for astronauts on the space station, and think about how we might want to use genetics to make that even better. So if you think about something like a tomato plant, where you spend all this energy growing this plant just to get these tomatoes, maybe we want to figure out ways to try and make the rest of that plant usable too. Maybe we want to use genetics to think about ways to make super composting microbes so that we can actually turn that plant material back into usable material that can grow another plant much faster. So there are lots of different organisms that we have questions about how microgravity is going to affect them, especially on these deep space missions that we want to start trying to answer now on the International Space Station. Now, there are a lot of challenges that come with spaceflight, but there are a lot of really cool opportunities as well. So this is one of the things that is near and dear to my heart, is cell culture. So we are able to grow cells here on the ground in a dish, but they kind of grow like a flat cell pancake because gravity keeps them sort of flat down to the bottom of the dish. But if you think about us, we grow in 3 dimensions. So if we want to try and grow cells up in space, we can actually use that microgravity to our ability to grow these different kinds of 3D cell cultures to better replicate how our own cells grow. We can also do things like improved crystal growth and crystal structure analysis up in space. So if we want to study the shape of a protein, one of the ways that we often do that in the lab is to crystallize it and then to look at that structure of the crystal to tell us more about the original structure of the protein. And as you might be able to imagine, when you're in this microgravity environment, you have opportunities to grow slightly different types of crystals that don't have the confines of gravity. So you can do very different things in space and use that microgravity environment as an opportunity to answer questions that you wouldn't be able to answer down here on Earth in our gravity-filled environment. So through the Genes in Space program, students have taken advantage of all of these different opportunities and challenges to come up with new biological questions and innovations. So again, we started the Genes in Space program back in 2015, and this is just a few of our winners here. So we started in 2015 with Sophia. So we're going to talk about her project in just a minute, but she was really interested in looking at how the immune system responded to the microgravity environment. We also have Julian. Julian was really interested in aging in microgravity, so he decided to write a proposal that would study the telomeres, the protective end caps on DNA that we know change with aging. And all the way up to last year's winners, Finsim and Yuji. So Finsim and Yuji were really interested in studying circadian rhythm genes and neural expression and neural cells up in space. So we've had a wide variety of experiments that have both been proposed and have now been launched up to the International Space Station. So there are so many different things that you can try and actually go and ask. And again, I mentioned Anna Sophia. So Anna Sophia was our first Genes in Space contest winner, and she was a high school student who had wanted to be an astronaut since she was a child. So when she saw the Genes in Space program, it seemed like a great way to combine her love of biology and her love of space and ask really cool questions. So Anna Sophia really wanted to design an experiment that would look at the immune system in space. But she wanted to use polymerase chain reaction, or PCR, to be able to amplify some regions of DNA. But we didn't know if PCR would work in space. It hadn't actually been done in space before. So Anna Sophia designed the first experiment to run this really integral molecular biology technique up on the International Space Station and showed that yes, in fact, we could do PCR up in space. So really, this amazing technique was run for the first time by astronauts, designed by her, and was in fact so important and successful that she became the first author of the published paper on this experiment, which is just really cool that as a high school student, she was doing world-class, space-class, publishable research, which is really, really fantastic. So again, I want to come back to sort of the basics of the competition here as we dive into what applying for this even looks like. It's open again in students from grades 7 through 12, and you can work alone or you can work in a team. But all you need to do at first is have a great idea. All we are going to ask you to do in your proposal is tell us 4 main things. So first of all, you need to identify a cool space biology question. Then you have to have a great hypothesis to go along with it. You need to tell us what your experimental design is that is going to use polymerase chain reaction to try and test that hypothesis. And then you have to justify why it has to happen up on the International Space Station. Why do you have to do this experiment up in space? Why can't you just investigate this down here on the ground? So there are lots of different types of questions that you could ask. Things like, how do we keep astronauts' muscles strong in microgravity? How can we create better foods for these astronauts? How does microgravity affect your immune system? Lots and lots of questions. And not just the final winner is going to get something out of this program. So we do have one final winner or winning team whose project goes to the International Space Station, but along the way we also have 5 finalists. So these 5 finalists are going to actually be paired with mentors and present their project at the International Space Station Research and Development Conference. We have 10 honorable mentions. We have 5 junior scientist awards. So these are for middle school competitors. And then we also have 5 Constellation Awards. So these are for the schools that submit the most applications. So there are 25 different ways to sort of win and participate in this contest beyond just learning about all of this cool space biology by writing up your proposal. So these awardees will be announced in May. And then those 5 finalists, oh, and I should mention that all 25 of these awardee winners, their teachers will get a DNA discovery system, which is this mini PCR and the electrophoresis and a pipette that will allow you to do molecular biology experiments in your classroom, which is pretty cool. So these awardees will be announced on May 21st. And then, as I mentioned, we have 5 finalists, and these 5 finalists will be paired with mentors. Now, these mentors are amazing Harvard and MIT graduate students, and they're going to help these 5 finalists really develop their proposal from just these 4 questions that we asked you to fill out in the application into something that they can actually present to a board of scientists to justify why their experiment should be the one that actually goes up to space. Now, these are amazing mentors. They develop really great bonds with these students and just help them all along the way. These are wonderful, wonderful people who I feel lucky to have had the chance to meet. And students really develop sort of lifelong relationships with these mentors beyond just this competition. And so those 5 finalist teams, with the help of their mentors, will actually go to the International Space Station Research and Development Conference to present their work to a board of esteemed judges. Now, these judges include people from Genes in Space itself, Math for America teachers, NASA scientists, And every time that I have been there, there has also been an astronaut in the audience. And it is a rare opportunity for anybody to get to present their original ideas to an astronaut. I'm jealous of that opportunity that I don't really get to do it. And so it's a really phenomenal experience for these winners or for these finalists to present their ideas, their research, and their experiments. And then the winner will also be announced at that conference in August as well. Now, once that winner is announced, they will go on, or either that individual or the winning team will go on to space biology camp. Now, space biology camp changes every year depending on the actual project that has been proposed, but the idea is that the winners will get to work with their mentors and scientists from NASA to actually really build the protocol that will be done in space. So they will work on the actual physical reagents, they will work on the physical things that are going to go to space, and it's kind of cool because these students get to write the protocols that the astronauts are going to do up in space. So it's an opportunity for you to tell an astronaut what to do in the name of science, which I think is really exciting. And after space biology camp, once this experiment is totally prepped and ready to go, then it is launched, and the winning team and their teacher get to go travel down to Cape Canaveral to actually watch it launch to the space station, which is really just a once-in-a-lifetime opportunity to get to do that. So I'm jealous I haven't gotten to go, but these students really take so much away from it. And this could be you. You could be at Cape Canaveral watching an experiment that you designed launch to the space station to be done by astronauts. So I really love this quote from Michelle. She was one of our 2018 Genes in Space winners. So she said, "I think the best thing about the competition is it doesn't really end just with a trophy and a medal and you go home just feeling proud of yourself. You get to actually take this experiment and implement it into a real life situation and actually get it carried out in space." And that, I think, is totally the coolest part of this contest, is that you are doing real science and you are sending something to the space station that is going to have an impact on all of the molecular biology that is done there for years to come. So you are really doing real science that is adding to the body of work and adding to what we know about life in space. And again, I just want to put forward that there is one winner and there are these 5 finalists and 25 awardees, but there are also hundreds of students who write in these proposals who really get something out of this by learning about space biology and getting to really become an active part of that space biology community. So even if you're not the winner, just writing up this proposal and becoming a part of this community is something that can have an impact on you and your sort of ability to communicate with research and do research for years to come. So it's a really, really great opportunity. And the 2020 contest is now open. You can submit your proposals right now. It opened up in January. And I am so excited to announce to you right now that in fact we have extended the deadline for your proposals until April 25th. So you have a whole month from now to come up with your ideas, write your proposals, and submit them. So you have plenty of time to do this. We've extended it so that you have even more time, and I am really excited to be able to tell you that yes, now you have until April 25th to get this working. So this is our updated timeline here. So contest opened on January 13th. Submissions are due April 25th. We will announce the finalists on May 21st, and then the winner will be announced at the ISS Research and Development Contest conference on August 6th. And then finally, we will launch to the International Space Station in spring of 2021. So again, we've launched 6 of these experiments so far. There's another one going up soon. You could be at Cape Canaveral watching your experiment launch up to space, and I just can't think of anything cooler than that. That's really exciting. So this is what the application form actually looks like. So here's your submission form. You're going to fill out those 4 questions that we've talked about before. And we have a lot of ways to try and help you think about and form these questions. So first things first, you got to think up a cool open question. So we have some starting points here that you might want to think about. Perhaps you're really interested in detecting life. How can we use PCR to detect new life forms? Or maybe you want to think about microbial monitoring. Maybe you want to come up with a cool way of looking at those microbes that live on the International Space Station that might affect how we're traveling into deep space. Or maybe you just want to look at how DNA behaves up in space. How does it repair damage? How does it respond to this microgravity environment? So lots of cool ideas. And we've tried to come up with some resources online to help spark these ideas as well. Once you come up with that question, now you have to formulate your hypothesis. So what do you think about that question? And then your experimental plan. So how are you going to actually test your hypothesis and answer that question? And part of your experimental plan has to incorporate PCR. So we've been talking about this thing PCR a lot. If you haven't heard of it, we're going to dive deep into it in just a second. But the idea here is that you have some kind of complex DNA sample, you're interested in a small region of it, and you're going to make lots and lots of copies of that small region in order to study it. So your experimental plan has to incorporate PCR. And then finally, again, you have to tell us, why does this need to happen up on the International Space Station? Why can't this just happen down here on the ground? Why do we need to send it actually up into orbit? So let's come back to PCR. So as I mentioned, you have a complex DNA sample, but you're only interested in one little piece of it. So let's say, for example, that your complex DNA sample is the entire human genome, 3.2 billion bases long, and what you want to do is you want to study one specific gene that's maybe 500 base pairs long. How are you going to find that specific piece and then study it? Well, PCR lets you make lots and lots of copies of that piece, so it's easier to find and easier to study. So it works a little something like this. So we're going to use an example here where I want you to imagine that instead of your DNA, your human genome, being 3.2 billion bases long, let's pretend that instead it is written out on sheets of paper 500 bases at a time. So you get lots of printed out sheets of paper. If you were to do that, your paper would create a stack, or your genome would create a stack of paper 1/3 of a mile tall, which is just a little taller than the spire on One World Trade Center. So this would be a huge stack of paper. But you are only interested in studying a single page out of that giant stack of paper. So how are you going to find it? It's going to be really hard to sort through all those sheets of paper just to find that one that you're looking for. So we can use PCR to make lots and lots of copies of that one piece of DNA, that one piece of paper that you're interested in, to make it easier to find. So it works a little something like this, where you have your single sheet of paper, and then you use your PCR machine like a photocopier to now make 2 sheets of paper. You run both of those back through the photocopy machine, and now you have 4 sheets of paper. You take those, you run them back through the photocopy machine, and now you have 8 sheets of paper. So you are exponentially amplifying this region that you are interested in over and over and over again. And we do that typically in PCR for about 30 to 35 cycles, until at the end of that, you would have a stack of paper 63 miles tall. So imagine that now you have that original 1/3 of a mile that's your whole genome, and 63 miles of just the piece that you are interested in. It is going to be a lot easier to find that piece that you're interested in now than it was before. And that's exactly what we're doing with DNA in PCR. So it looks a little something like this when we're doing this in a tube. So first of all, you have your complex DNA sample, and you're going to use heat to break the 2 chains of sides of DNA apart, and then you are going to introduce something called primers. So these are short little pieces of DNA that specify the beginning and the end of the region that you want to amplify. So that is your region of interest. Now you're going to cool your reaction down a little bit, and those primers are going to bind to just that region of interest. Now that your primers are bound, you're going to heat your reaction back up just a little bit, and you're going to introduce something called a polymerase. Now this polymerase is a protein that is going to bind to those now double-stranded regions designated by your primers, and it is going to copy those 2 strands of DNA using all of these additional DNTPs as building blocks. So now you started with just 1 piece of DNA, and now you have 2 pieces of DNA, and you're going to do that over and over and over again. So we have lots and lots of resources for you to learn more about things like PCR and to learn more about the competition online on our Genes in Space website. So this is genesinspace.org. We have a bunch of resources. So first up, we have a bunch of video resources. So we have lots of really great interviews with scientists who are actually running space biology projects. So here on the left, we have Dr. Joy Amasa running, again, one of my favorite projects, Veggie, looking at how we're going to feed astronauts as they go deeper into space. And then we also have a number of videos that look at the molecular biology techniques that you're probably going to want to use in your proposal. So things like PCR, really diving deep into the science of that. We also have a bunch of really great Genes in Space chats. So these are live chats on Facebook, and in fact, there is one tonight at 8 p.m. Eastern with our mentor and amazing MIT scientist, Holly Christensen. She's going to be talking about different types of PCR, but these are opportunities for you to ask all of your questions and get them answered in real time. So we have one tonight with Holly, again, on our Facebook page, which is just facebook.com/genesinspace. We're also going to have one next Monday with one of last year's finalists, Abhi, and then we're going to have 2 Meet the Mentors sessions in April. So make sure you go check out our Facebook page for those. And again, please join Holly tonight. She's going to be there to answer all of your questions about different kinds of PCR, as well as just writing up your proposal and your application. We also have prior submitted applications. So this is one of our finalists from a prior year or honorable mentions, so that you can get an idea of what a great proposal looks like. So you can go to our website. All of this is at the Learn page, which is genesinspace.org/learn. And all of these links are also in the description down below. So if you want to go and check out any of this information, you can go and look at it all right there. Should be very easy to find and search out. Now, before we wrap up and head into the Q&A session of this webinar, I just want to point out that this is one of our finalists from last year, our 2019 finalist, Abhi, and his project was reassortment of genetic material in the influenza A virus in space. Now, Abhi is a high school student from Clear Lake High School in Houston, Texas, and I think his project is a really great example of the fact that a lot of these ideas can really have real-world implications. So he was interested in studying viral reassortment in microgravity. His project asked whether or not viruses exchange genes differently in microgravity than they do down here on Earth. And while his project was focused on trying to help astronauts as they venture deep into space, the knowledge that we learn from those kinds of projects and those kinds of studies can be really applicable to those of us down here on Earth struggling with things like the novel coronavirus. So I think that Abhi's project is a really good example of the fact that the questions that these students are asking are real important scientific questions that can have huge implications for biology. And they're coming from middle and high school students. So that's not to say that your proposal has to tackle some of the big scientific questions and challenges that are facing our world right now, but it is to say that you can have real world impacts and do great work, all just by having a great idea and submitting a great idea. So Abhi, again, is going to be doing a chat with us on Facebook on March 31st that I think is going to be really, really interesting. This is next Tuesday, sorry, not next Monday. And I think it's just a great example of the fact that you can be asking questions that can really be helping lots of people. So I want to dive quickly into our Q&A session. We got some questions that came in before the session that I want to answer. So one of the first questions that came in is, how relevant must PCR be to our proposals? And it has to be very relevant. So we are really looking at genes in space. We're looking at how genetics can help us to answer these space biology questions. So your experimental plan has to include polymerase chain reaction. And again, we have lots of resources going, really diving deep into the details of PCR on our website. And another thing that I want to show here or highlight here is that you also want to think about, what are you doing after PCR? So let's say you want to study a gene involved in the immune system. So if you amplify that gene up in space, what are you going to do with it next? Are you going to try and sequence it? Are you going to try and visualize it in some way? Once you have that piece of DNA that you want to study, how are you going to study it? What is that next step going to be? So you want to think about PCR, but you also want to think about what comes after PCR. How are you building that experimental plan? Another question we got is, how many applications may one person submit? And the answer is just one. So you can submit either as an individual or as a team member. So make it count. But you can submit multiple years in a row. So if you have submitted in a past year, you can submit another one. You can just only submit one application for this year. Somebody asked, can we incorporate the use of test organisms? And model organisms are a really important part of biology, and you should absolutely think about model organisms, but you should also think about the ability to send those model organisms up into space. So you're probably going to be much more likely to send something like yeast or a little C. elegans worm and study that up in space than you are going to be able to study a moose up in space. So if you are going to be thinking about model organisms, we encourage proposals that have them, but you do want to be thinking about what are reasonable model organisms that you can study up in space. Somebody else asked, can DNA synthesizing for primers be done in space? At the moment, I do not believe that we have the ability to synthesize DNA primers up in space, but the cool thing is that you can design them down here on Earth and then send them up as a part of your experiment. So you can 100% design primers, you can 100% choose what genes you want to amplify, and you'll just send those up to space with your experiment if it is chosen. Somebody asked, what is the most important part of the proposal? And it's really hard to identify just one part. All parts are very important. It's really, the review of these is really holistic. We look at the entire application together. So there isn't just one magical part. You really want to think about all of the different parts of this application and really give all of them sort of your time and consideration. Somebody also asked, if you're unsure about putting something in your proposal, should you always just put it in, even if it has to get changed later? So it depends a little bit on why you're unsure of putting it in, but we don't really want you to worry about feasibility right now. We don't want you to worry about the cost of an experiment. We don't want you to worry about, things that are going to be a little hard. Again, you probably don't want to propose sending a moose up to space, but don't worry about the feasibility so much. We're really interested in your questions and your experiments and your proposals. So if you're not sure whether or not something is doable, if you think it might be right on that line, I would say err on the side of including it. Add it in, and we'll work that out farther down the line with your mentors about what you can and can't do likely on the space station. Let me check some of these other questions here. So somebody asked, what's the best way to integrate the use of PCR? And that's kind of up to you. So again, you want to make sure you're using PCR to study a specific gene or RNA or a number of different things, but you want to make sure instead of just asking or proposing, I'm going to study the immune system, you probably want to think about what gene is going to tell you what you want to know about the immune system. So if you want to know, say I used to study the heart, if you want to know how the heart responds to microgravity and use genes to look at it, you probably want to identify a specific gene and say, for example, does the RNA expression of MYH7 go up or down in response to microgravity? So you want to really sort of narrow in on what is that specific gene or RNA or nucleic acid that you are going to study in this experiment. So make sure that you can have a really broad idea, but make sure you narrow it down to something that's actually feasible and testable. Some other frequently asked questions. Can I form a team with students from different schools? Absolutely. If you're at one school and your friend is at another school and you want to write a team proposal together, we 100% encourage that. Some of our 2018 winning team was made up of students from different high schools. If you're homeschooled, can you participate? Absolutely. So we welcome submissions from homeschooled students. And again, do you need biotech equipment or access to a lab to enter the competition? No. All you need is a great idea. You do not need to do any experiments up front. You just need to ask a cool question and give us some ways that you would want to test that up in space. But you do not need any equipment. All of this is just written at this stage, and you can do it all just by submitting your application online. And again, that is at our genesinspace.org website. So thank you so much for joining me today to chat about genes in space. I so appreciate that a bunch of you showed up. I saw about 60 people on the stream at one point in time. So I hope you all submit to the Genes in Space contest. Again, genesinspace.org is where you can find out all of this information. We have tons of resources to help support you. Remember to stop by our Facebook Live tonight, and you can chat with Holly about all of your questions about writing up your proposal. And then finally, I did want to just give a final mention. Again, Mini PCR Bio is one of the sponsors of the Genes in Space competition. And if you have questions, as I believe many of us do, around COVID-19 and how genetics might actually be able to help us investigate and work on and solve the problems that we're all really being affected by right now, I'm going to be leading another webinar next Monday at 3 p.m. Eastern, noon Pacific time, to talk about that and how we can use genetics to try and learn more about COVID-19. So if you're interested, you can sign up and join us for that as well. That's going to be on the Mini PCR Bio YouTube page. So once again, I just wanted to say thank you so much for joining me today. It was awesome to get the chance to tell you about this amazing, amazing contest. I hope that you all submit all of your questions, and we'll see you again soon in the future. Have a wonderful rest of your day.